welcome to our channel. This is Science Lectures. Thanks for being with us. Please consider subscribing and like our videos. Thank you. So today we are going to specifically look at the bacterial cell, and the bacterial cell falls under the kingdom. So we need to know the kingdom where it falls under. So it falls under kingdom Monera. And under this kingdom Monera, we have two basic things. That is bacteria. We have bacteria in under Monera. Then we also have cyanobacteria. Cyanobacteria, which is also known as blue-green algae. However, we know that we have also other kingdoms of life. And other kingdoms include the animal kingdom Animalia, where we have all animals from sponges to flatworms, then finally to man. Those are some of the examples in kingdom Animalia. Then we have another kingdom, which is kingdom Plantae, where we find a variety of plants. Then we have another kingdom, which is fungi, kingdom Fungi, where we have yeast. We know yeast is single-celled. Then where we have yeast as part of the examples. We have mushrooms, yeast, mushrooms. Then the mold also is part of the fungi kingdom. Then finally, we have kingdom Protista, where we have protozoans and algae. Under the protozoans, we'll see flagellates, ciliates, and the rest of them. So all these kingdoms will be covered within the session of this video. So you keep with us on the channel. We'll be discussing kingdom Animalia, kingdom Plantae, kingdom Fungi, kingdom Protista, and today we are looking at bacteria under kingdom Monera. So this is the basic structure of our bacterial cell, and we are going to give the different parts and the functions of each of the parts of this cell. So we are going to start with what we call the appendages, extensions of the cell. So our first appendage is the flagellum. Then we have some other structures there, some other extensions, cell extensions. This extension is called the pilus. Then we also have a black extension there, which is called the fimbriae. Then we have this covering, the outermost covering, which is the capsule of the bacterial cell. Then the second covering inside, which is the cell wall of the bacterial cell. Then finally the innermost covering, which is the cell membrane of the cell. So that is our cell membrane. And these three, the three outer membranes, that is the capsule, the cell wall, and the cell membrane. All at large form what we call the cell envelope. So they are basic parts of what we refer to as the cell envelope. So let's name some other structures inside the cell. We have that structure inside the cell, which is called the mesosome. Then we have the DNA, the chromosomal DNA of the bacteria. chromosomal DNA. Then we have another type of DNA, which is called the plasmid. Plasmid, the form of genetic material also inside the cell. Then we have the cytoplasm, the fluid where the organelles are suspended. Cytoplasm. Then we also have some ribosomes inside there. Ribosomes. Ribosomes. So we are going to have a look at each of the parts of this bacterial cell in detail and give the functions of each of the parts, because we need to have detailed information about each of the parts and the functions it carries out. So we'll start with the flagellum. The longest cell extension, which is called the flagellum. And the basic role of the flagellum is it aids in what we call locomotion. So movement of bacterial cells is aided by the flagellum. That tail-like structure. So this flagellum obtains energy in form of ATP, and the energy, hydrolysis of energy in form of ATP, produces the phosphates that provides energy for beating action. The beating action of the flagellum for locomotion of bacterial cells. And locomotion is very important because if a bacteria wants to move away from maybe a toxin, or it wants to move to look for some nutrients, it will be able to carry out this. However, some bacteria have the location of the flagellum differing. You will find some bacteria having the flagellum on one side of the bacteria, others having the flagellum on both sides, others having the flagella on one side, then all over the bacterial cell. So when we have one flagella on one side, that is what we refer to as monotrichous. Then when we have many flagella on one side of a bacterial cell, monotrichous, for example, in Vibrio cholerae, bacteria like in Vibrio cholerae, that bacteria that causes cholera is monotrichous. Only one side having a high concentration, having one flagella. Then if we have two flagella on both sides of the bacteria, that is what we refer to as amphitrichous. So some bacteria are amphitrichous. Then if we have them towards only one side, this is what we refer to as lophotrichous. Then if we have flagella surrounding the entire bacterial cell, this is what we refer to as peritrichous. For example, in the bacteria E. coli, Escherichia coli will possess peritrichous, will have flagellum all over the body. And we say the basic function of this flagella is for locomotion of the bacterial cell. However, they are provided energy in form of ATP in order to carry out their locomotory function. So this is basically what the flagellum does. So let's have a look at another cell extension. We are going to start with cell extensions. Let's have a look at the pilus. The pilus and the fimbriae. The pilus. The pilus is the red structure that is bigger in size, that is thicker. The pilus is thick, is longer, is thicker and longer as compared to the fimbriae. In some books, the pillars of fimbriae are considered to be the same. However, they are kind of different. The pillars are many. They are many in number. That is what we should know. They are many in number. They are thick and longer as compared to the fimbriae. If we are to have a look at our fimbriae, the black ones, our fimbriae are short. They are shorter as compared to the pillars. They are shorter. Then they are thin. They are thinner. They are not thick. Then finally, they are few in number as compared to the pillars. They are few. Sorry, they are many as compared to the pillars. The fimbriae are many. Then that means the pillars are few. The one in red, they are few. However, we also have another basic difference between the pillars and the fimbriae, and that difference is in the function. The fimbriae will serve a function of adherence onto surfaces. So they are the ones used to adhere onto surfaces. For example, let's imagine we are having a catheter in a patient. For the bacteria, for example, Pseudomonas, to adhere onto the catheter surfaces, they will need what we call the pillars. It will be done with the aid of the pillars being present. So basically, the aid of the fimbriae present. So basically, the fimbriae are used for adherence onto the surface. However, the pillars are used for conjugation, a form of sexual reproduction we refer to as conjugation. Conjugation involves the transmission of a plasmid, transmission of the contents of a plasmid from one bacteria to another bacteria. So this conjugation leads to recombination in the genetic composition of this bacteria. But in our lecture today, we'll be seeing what conjugation is in detail. So the fimbriae, we've said basically they are used for adherence and attachment onto the surfaces. However, they are shorter, thinner, and many as compared to the pillars. The pillars are thick, few, and longer in length. However, these pillars, we say they do a basic role in conjugation, but they do still have another difference. And where does the difference lie? The pillars is formed, synthesis of the pillars is aided by the plasmid. That extra chromosomal DNA. Then synthesis of the fimbriae is aided by the chromosomal DNA. Chromosomal DNA. So that is it basically for the pillars and fimbriae as other cell extensions. So in summary, we have three cell extensions. We have the flagellum, we have the fimbriae, then we have the pillars. The flagellum, we say it is used for locomotion within the cells by the aid of ATP. Of course, it needs energy in form of ATP, and we say it can be distributed in different ways according to different bacteria. Then we also have what we call the pillars, which is used in conjugation, and it is synthesized with the aid of the plasmid. Then finally, we have what we refer to as the fimbriae, that is used for adherence onto. So let's have a look at another basic component, the one we call the cell envelope. And we say the cell envelope has three basic parts. It has what we call the capsule, then followed by the cell wall, then finally inside we have the cell membrane, the cell membrane. So this cell envelope, we are going to start off with the capsule, and we need to know the importance of the capsule. Most bacteria have a capsule on as the outermost covering. So this capsule, the importance of the capsule is it works as a virulence factor. Virulence factor. What do we mean by working as a virulence factor? It prevents or inhibits, prevents phagocytosis. Phagocytosis by the host cells. Let's say we are having monocytes or macrophages. Let's say in the human body or the body of any other animal like fish, it possesses macrophages or monocytes. When these macrophages or monocytes, if they want to attack the bacterial cell, that coating of the capsule protects the bacterial cell from phagocytosis. That is how it works as a virulence factor in preventing attack from the outside by the host cells. Then the cell wall has many functions. It will provide shape to the cells. It is basically for shape. Then it will provide support to the bacterial cell. Then it prevents bursting. That means it resists what we call osmotic pressure. If we have osmotic pressure being high, where after entry of water into this cell, the bacterial cell will resist increase in Tucker pressure, so it ends up protecting the cell from bursting. Then another function of the cell wall, basically shape, support, and osmotic pressure. However, the cell wall of bacterial cells is a peptidoglycan layer. Peptidoglycan is made up of peptidoglycans. And peptidoglycans, this layer can be, this cell wall can be either of a gram-positive bacteria or of a gram-negative bacteria. So if we are having bacteria that are gram-positive and gram-negative, they will differ somehow in their peptidoglycan layers. So gram-positives will have a thick peptidoglycan layer, whereas gram-negatives will have a thin peptidoglycan layer. And we'll see how this one is relevant when we do gram staining. Gram staining relies on the principles of the thickness of the peptidoglycan layer in gram-positives and in gram-negatives. So this one will go in details in the technique for staining, the staining technique. So basically that is what our cell wall is doing in the cell. Then the cell membrane, we know the cell membrane is a semi-permeable membrane. So that means it will will allow entry of substances at different extents. So entry of substances at different extents will allow some to pass freely, will permanently limit entry of substances, and will allow entry of some to a smaller extent. So basically that is our cell envelope. The capsule, the cell wall, the cell membrane. Capsule, virulence factors, preventing phagocytosis by the host cells, like macrophages and monocytes. Then the cell wall, basically for shape, support, osmotic pressure. And again, we can use the cell wall for classifying bacteria, because a cell wall provides shape. So if the cell wall is round, if a cell wall is rod-like, and if the cell wall, let's say, is a rod which is bent, we can use the cell wall for classifying bacteria. Then it can resist increase in osmotic pressure. Then we know that now the cell wall contains the peptidoglycans, the peptides, and the carbohydrates. But these peptidoglycans are either for gram-positive bacteria or gram-negatives. But we say peptidoglycan layer in gram-positives is thick, while the peptidoglycan layer in gram-negatives is thin. And finally, the cell membrane that helps in the transportation of materials into and out of the cell. So that is the cell envelope. We are done with the appendages and we are done with the cell envelope. So let us look at the internal contents of the cell. Our first internal component we'll talk about is the mesosome. The mesosome does a role in respiration. So this one, not respiration, it will do a role in ATP production. So this one does metabolism. The mesosome will do metabolism. Remember prokaryotic cells. This bacterial cell is a prokaryotic cell. It doesn't have what we call the mitochondria. So absence of the mitochondria, it will need an organelle that is used in synthesis of ATP, and that is the mesosome. However, you should also know that the ATP produced is always less as compared to the ATP produced by the eukaryotic cells. In other words, prokaryotic cells, this bacterial cell, will have only glycolysis taking place. So it doesn't have the Krebs cycle, that right, carboxylic acid cycle. So that is the importance of the mesosome. Then ribosomes in this bacterial cell are 70S. There are 70S ribosomes. These ones are for protein synthesis. So ribosomes are sites for protein synthesis within the cell. So the cell will be able to synthesize proteins. Then another internal organelle is the chromosomal DNA, the DNA of our bacterial cell. And the DNA of this bacterial cell is will help in replication. It stores the genetic information on which we'll have protein synthesis. So for protein synthesis to take place, it requires some form of information stored, and that information is stored in the genetic material of this bacteria, and which is the DNA. However, the DNA of this bacterial cell is circular. Number one is circular. And second thing we need to note is that it is found in a single chromosome. A single chromosome of circular DNA. That is how this DNA is arranged. So a single chromosome of circular DNA. That is the DNA of our bacterial cell. Ribosomes, mesosome we've seen. Then it also has a cytoplasm. Cytoplasm where metabolic reactions are taking place. Different metabolic reactions are taking place, and where the cell organelles are suspended within the cytoplasm. Then finally we have a structure known as the plasmid. The plasmid is very, very, very vital for these bacterial cells, because this plasmid is like an extra chromosomal DNA. It is an extra chromosomal DNA, and this plasmid can have genes, special genes that are helpful in survival of the bacteria. For example, genes for antibiotic resistance. Antibiotic resistance. So some bacteria are resistant to antibiotics. For example, some bacteria are resistant to penicillins. But one of the factors that are driving antibacterial resistance is presence of this extra chromosomal DNA we refer to as the plasmid. The plasmid can be transferred from one bacterial cell to another with the aid of the pilus. With the aid of the pilus we gave the function of the pilus there. So genes for antibiotic resistance can be encoded within that pilus we are seeing there. So the cytoplasm, the chromosomal DNA, and so that is it with the basic function of the different organelles within this bacterial cell.